So good day everyone. Today we're going to metabolic pathway 2 as now is my course outline here in my school. And then this metabolic pathway 2 is talking about amino acids, how amino acids and protein, how they are being processed in the body, their metabolism. So now before we get into the metabolism of this amino acid, we'll first be talking about some basic information and things about proteins we need to know, and some degradation and sequencing of proteins and of amino acids that we'll be going through before we eventually reach and go into the metabolic pathway proper. So yeah, let's begin the course. Now, the overview of proteins. Proteins are seen to be made up of amino acids. Now, these amino acids are connected together by peptide bonds, forming polypeptide chains, and these polypeptide chains could be as long as being up to 1,000 amino acid residues in this polypeptide chain. Now, these amino acids are linked together by peptide bonds that I said before, and these peptide bonds are formed between the carboxyl group of the first amino acid and the amino group of the second amino acid. Now let's look at this here. We see how the peptide bond is formed between the carboxyl group, that is CO, and then the amino group of the second amino acid, and then that is how it goes. So conventionally, in the structure of a protein, or in the primary structure of a protein, the first functional group that is seen is the amino group, is the amino functional group, and then the functional group at the end of the chain is the COO group. So now let's move on. Now, structures of proteins: we have the primary structure, we have the secondary structure, we have the tertiary structure, and the quaternary structure. Now, the primary structure is made up of the unique and precisely defined amino acid sequence. Now, for every protein, there's a specific amino acid sequence they have. Now, coming to the secondary structure, we have two secondary structures. We have the alpha helix, or the alpha helix, and the beta pleated sheets. Now they are formed by hydrogen bonding between amino acids present in this amino acid sequence. Now, Pauling and Corey predicted the existence of this secondary structure back in 1951. Now, for the alpha helix, it is a rod-like structure. That is formed as a result of hydrogen bond between the amino acids present in the polypeptide chain. Now, the hydrogen bond is formed between one amino acid in the chain and then 4 after it in the chain. That's how the hydrogen bond is being formed. And now, in the alpha helix, each amino acid is related by related by distance of 1.5 angstrom, which is equals to 0.15 nanometer. Now, for this alpha helix, we have the right-handed and the left-handed. Now, the right-handed is seen to be like clockwise when viewed down the axis of the helix, and for the left-handed, it is seen to be counterclockwise when viewed down the axis of the helix. Now, the right-handed is seen to be more common is more seen in proteins. Now, essentially all alpha helixes are found in proteins are right-handed. So alpha helix is more the right-handed alpha helix is seen more in proteins than the left-handed helix. Now we come to the beta pleated sheet. Now, in this beta pleated sheet, the polypeptide chain is nearly fully extended. 2 or more beta strands connected by amino to carboxyl hydrogen bonds come together to form beta sheet. Now, the distance between the adjacent amino acids is 3.5 angstrom, which is equals to 0.35 nanometer, and then it's also stabilized by hydrogen bonds. So something we should note among the secondary structure is that the relations between the amino acids is as a result of hydrogen bonds being formed in them. Now let's come to the tertiary structure. The tertiary structure refers to the 3-dimensional structure of the entire protein, of the whole protein. Now, it also defines the steric relationship of amino acids which are far apart from each other. Now, amino acids that are far apart in the polypeptide sequence that resides in a secondary structure, whether the alpha helix or the beta plated sheets tend to interact with each other, and then they interact with each other by electrostatic bonds, hydrophobic bonds, and van der Waals forces. So they basically interact with non-covalent interactions. And now the tertiary structure acquired by the native protein is most thermodynamically stable. Now this is an example of the actual structure of the 3D foldings. Now we see the alpha helix, here, and then we see the secondary, we see the beta sheet here. So the alpha helix and the beta sheet can be found in a polypeptide chain, and be found in just one polypeptide chain. Now we come to the quaternary structure. It talks about when there's interaction between one or more polypeptide chains, and then this forms the whole quaternary structure. But now when just one of these polypeptide subunits is dissociated from this quaternary structure, the protein loses its functions. Now this quaternary structure is the forces that keep the quaternary structure together is the hydrogen bonds, electrostatic bonds, hydrophobic bonds, and van der Waals forces. And then from the diagram we have here, or for the figure we have here, we see we have 3 polypeptide chains. We have polypeptide 1, polypeptide 2, and polypeptide 3. Now this is just an overall showing us everything about the structure of the proteins. Now we see the primary structure here. Going down, we see the secondary structure, the alpha helix and the beta plated sheet, and then we see the tertiary structure, the 3-dimensional structure of the entire protein, and then we see the quaternary structure showing different interaction of polypeptide chains, and here we have 4 of them. So let's move on. Now depending on the number of polypeptide chain, they can be called either dimer, tetramer, and so on. And then depending on whether the polypeptide chains are the same, we can then introduce homo and hetero. Now let's say we have 2 polypeptide chains and both of them are of the same amino acid sequence. We call that now hemodimer. But now in the case of, let's say the 2 polypeptide chains are different, they contain different amino acid sequence, it then becomes an heterodimer. Now we go example of the hemoglobin. It has 4 polypeptide chains, 2 beta chains and 2 alpha chains. Now from this you can now say it has a heterotetramer quaternary structure. So that is that for hemoglobin. Now we go to the amino acid sequencing of proteins. The sequencing of proteins is done by Edman degradation, and the Edman's reagent is phenyl isothiocyanate, that is PITC, or you can just call it PTC to remember PTC. Now it forms a covalent bond with the N-terminal amino acid, that is like the first amino acid in the primary structure. It forms a covalent bond with it, and this is called labeling. Now after this is formed, this compound that is being formed between the phenyl isothiocyanate and the first amino acid is then cleaved, or the bond between the amino acid that is bounded by the phenyl isothiocyanate and the next one after it, that peptide bond is broken down by anhydrous acid. An example of this anhydrous acid is sulfuric acid and HCl. Now an anhydrous acid is an acid that water hasn't been added, which means it's not diluted and is in its most concentrated form. Now after it has been detached, it is then identified by chromatography, and then the Edman reagent will react with the second amino acid, and then it goes on and goes on. Now this Edman's degradation is it's seen and is best done for 10 to 30 amino acids. Now above this number, sequencing by Edman degradation becomes not so reliable, and then this chain has to be broken down by hydrolysis. Now this hydrolysis can be done by either endopeptidase or by chemicals. Example of the endopeptidase that can be used is trypsin or chemotrypsin, and for the chemicals it can be cyanogen bromide, and you can just write it as CNBr. Okay, so now this is just a figure of what exactly happens in the Edman degradation. The phenyl isothiocyanate reacts with the first amino acid, forms a compound there, and then that is labeling. And then now is the peptide bond between alanine and glycine is broken down by the anhydrous acid, and then this is formed, the PTH and alanine, the amino acids identified by chromatography. Yeah. Now, so for trypsin, now trypsin, chemotrypsin, and cyanogen bromide, now they act in a way to cleave different amino acid peptide bonds in a way. Now for trypsin, the peptide bond formed between lysine or arginine and the next amino acid is cleaved. The peptide bond between lysine, let's take for example lysine, the peptide bond between lysine and let's take the next one is alanine, is being cleaved. It's being cleaved by this trypsin. So it's being cleaved by trypsin. So now the first chain will end with trypsin. The first chain will end with lysine, and now the second chain from the hydrolysis will begin with alanine, and then also arginine. That's what happens. Then chemotrypsin acts on the peptide, and then it phenylalanine, thyroxine, tryptophan, and leucine. Then for cyanogen bromide, it attacks the methionine residue. So yeah, that is how that works. And lastly, we'll be talking about disulphide bonds. Just a cysteine residue in a protein forms the disulphide bond, and then it acts as a way to stabilize these proteins. Now, example of a substance where we see this disulphide bond is insulin. Now, in insulin, there are 3 disulphide bonds, and insulin is also a peptide hormone. So that we see 3 disulphide bonds there, which we see two inter disulphide bonds between chain A and chain B, and 1 intra disulphide bond just on chain A. So yeah, that is all for this class, for this lecture. And then moving on from this, we'll now be going into the major metabolism of these amino acids and all that in the body. So that will be the next thing we'll be going into, the urea cycle and all that. So yeah, for now, let's digest this video, let's get to understand it and all that. So that is it for today. Yeah, thank you, and don't forget to like and subscribe. And also you can use the comment section on the ways we can improve on the YouTube channel or things you feel we need to add. Thank you.